everyone, and welcome back to Neuropsychology. So we are going to continue talking about chapter 17, which is about the connectivity, and we're specifically going to talk about Downer's commissure lesion experiment, so when the two hemispheres were disconnected. And we're going to talk about some connectome studies of how brain regions are connected. And we are going to finish off this video with Liebman's theory of apraxia. Okay, so let's start off with Downer's experiment. So John Downer investigated disconnections and their behavioral effects. So let me get my mouse. Here in this picture you have a normal monkey, and then here you have two monkeys, or they could be the same monkey. But what he did first was he severed or cut all connections between the two hemispheres. And then on top of that, he also took away the left amygdala. So he cut everything between the two hemispheres and he cut the amygdala. Oh, and these pictures are very schematic pictures where we're looking down upon a brain of a monkey. So that's why they're a little different than the human brain. So again, the midline, the commissures were cut, and the left amygdala was cut. And as you guys all know, the amygdala is very important for fear responses and emotion and aggression and stuff like that. Okay, so next he covered the eye of the animal, either the right or the left, and then presented the other eye that was not covered with objects. So if the objects were presented to the eye that is ipsilateral to the hemisphere with the removed amygdala, the animal appeared tame, even if the objects were scary objects that normally frightens monkeys. So that's this example. Here you have the taken out amygdala, all the commissures are cut, so there's no communication from left to right. If he covered this eye, and he presented items here, the monkey was tame. So it has something to do with information going into the eye and not being able to cross over and not having access to a amygdala. And if these objects were presented to the eye contralateral to the removed amygdala, so in this case, so the removed amygdala is here, so if the items were presented to this eye, then the animal would express his usual and species-typical response to threat, so he would appear a little bit more wild. Okay, so why is that? So here, if the information goes in here, if he sees threatening stimuli, the information cannot cross over, but it does have access to the amygdala, since the amygdala on this side is intact. So, for an animal to display species-typical response to a visual stimulus, aka acting a little wilder, or when he sees a threat, the information must project from the eye to the visual cortex, and then it goes through the temporal lobes to the amygdala. And then from the amygdala, it will go to the brainstem and then to the frontal cortex. Okay, well, I just told you guys a lot, so let's repeat that again. So when an animal sees a threatful stimulus, information goes from the eye, it goes then next to the visual cortex, then it goes through the temporal lobes to the amygdala, and then to the brainstem and frontal cortex. So information goes to the back of the brain, and then it goes through the temporal lobes to the amygdala, and then to the brainstem and frontal cortex. So these connections activate respectively autonomic responses, movements, and facial expressions. So when the commissures are disconnected, so when the two hemispheres are not able to communicate, visual information from one eye can project only to the ipsilateral hemisphere and not to the contralateral hemisphere. So if that hemisphere contains an intact amygdala, the behavior will be normal, aka he will show a little bit more aggression, so the monkey will look a little bit more wild. And if the amygdala in the hemisphere is not intact, the visual information will be disconnected from the motor systems, and the animal will not elicit any typical behavior. So he will just be kind of mellow and not threatened by the stimuli whatsoever. Okay, so had these commissures not been cut, would this experiment have worked? What do you think? No, it would not have worked. So why would it not have worked? Because if the commissures are still intact, the information from one hemisphere can cross over to the other, and each eye would thus have access to an intact amygdala, even though one is taken out. So the two hemispheres will communicate with one another. Okay, so definitely remember everything about Downer's experiment. Okay, so let's move on to cortical networks and hubs and connectome studies. So connectome studies demonstrate a large-scale neural network or networks that technically underlie very complex cognitive operations. And these studies seek to unravel neural networks architecture and how networks relate to cerebral functions. So often we use MRI for this, where we measure, where we get MRI or diffusion-weighted images, and then from there on you can do analyses on it. So brain networks can be extracted using diffusion-weighted imaging. We talked about that in chapter 7. And on top of the diffusion-weighted imaging, you can do diffusion-weighted tractography analysis. So this is something you can see in the left image. So here you see these cool little tractography analyses of these white matter pathways. And then basically, if you do further analysis on this, brain networks can be described by comprising sets of nodes, or technically neuronal elements, and edges. And the edges are their interconnections. So here you see all these little nodes, and all the little blue stripes would be their edge or their interconnections. So nodes interconnect with related nodes and cluster into functional modules, and that's what you see here. So here you have all the red dots are nodes, and here you see the nodes are blue or red or yellow. They're just the dots. So the nodes are the circles, and then the edges are the stripes between the circles. And the larger circle-shaped thingies are modules or functional modules. So all these nodes in this module have similar functions. So these modules have a lot of edges within them, and they also have some connections with other modules. And these connections are called hubs. So this is a hub, and this is a hub, and this is a hub. So provincial hubs are nodes that connect to primarily to similar nodes within a module. So here you see a provincial hub, and then you also have connector hubs that connect. Those are kind of nodes that connect to other connector hubs in other modules. So this is a connector hub. This is a provincial hub. What do you think that is? It is a connector hub. And what do you think that is? That is a provincial hub. Okay, good job. Okay, so we talked about diffusion-weighted imaging, but I wanted to go back. I copy and pasted a slide from a previous video, but I just wanted to reiterate over it. So what is diffusion-weighted imaging? So our brain consists of many water molecules, and these molecules move freely through the brain. However, they have some limitations, and they follow somewhat a path. So they can only go with fibers, and they cannot go through them. And why is that? It's because these fibers contain a lot of myelin, and myelin is fat, and water cannot go through fat. So if you follow the directionality of these water molecules, you can find the white matter fiber pathways. So here you see, in this image, you can see water molecules without any restrictions or without any boundaries or myelins. They can just go wee, they can go anywhere. And here you see the fatty or the axons with fatty myelin around it. So water molecules cannot go anywhere except for that way or that way. So they cannot go that way or that way. They have to go with the fibers. Okay, so let's move on to Liebman's theory of apraxia. So around the 1900s, Hugo Liebman, he published a bunch of papers demonstrating the importance of severed connections as an underlying factor in effects of cerebral damage. So he carefully analyzed the behavior of a particular patient, and then from there on, he predicted a series of new cortical disconnections that could account for his behavior. So he wrote a lot about the principle of disconnection, and particularly about the idea that some apraxias might result from disconnection. Okay, so there's a new term, apraxia. We did not cover that yet. So apraxia is the neurological disorder which is basically characterized by the inability to perform familiar or learned movements on command. So even though the command is understood and there's a willingness to perform this movement, the person cannot do this movement. So he reasoned that if a patient was given a verbal command to use the left hand in a particular way, only the verbal left hemisphere could understand that command. So here command, it first goes into the left verbal command center, what he called it. So to move the left hand, a signal would have to travel from the left hemisphere through the corpus callosum to the right hemisphere region that controls the movement of the left hand. Okay, so, well, that got confusing. So let's go over this again in this image. So when you hear a verbal command with your ear, it is first processed into the command center in your left hemisphere. Your command center will be always in your left hemisphere. So this information will then be sent to the motor cortex. But as you guys all know at this point, the motor system is 95% crossed, especially for your hands and digits. So to move your left hand, your information has to go over to your right hemisphere. So the motor cortex on the left will send it bloop, bloop to the other hemisphere, and that is when you can move your left hand. Okay, so if you have a severed commissure, or if you have no connections between left and right, so your corpus callosum is severed, you will hear the command. The command will go into the left hemisphere, your command center in the brain, and it will go to your motor center in the left hemisphere, but it cannot cross over. So the information can literally not get to the right hemisphere, so you will not be able to move your left hand. Okay, so little recap. So the person was, the same picture. So the person was commanded. So someone said, hey, move your left hand. That information goes to the command center, which is area 22, 39, and 40. And it is the command center is in the left hemisphere. Then it goes to the motor area. And if you have a severed corpus callosum, it cannot cross over. So you are not able to move your left hand because this information cannot go here. So big X here, information stops here. Okay, so if someone says, hey Puck, can you move your left hand for me? Again, information will go to the command center. Here. Command center on your left side only, left side of the brain. Goes to the motor area, your motor cortices, and normally it would cross over. You can move your left hand. However, if it's severed, that information doesn't go through, I cannot move my left hand. However, do you think with a severed corpus callosum that I can still move my right hand? Yes, I can, because the information is already in the left hemisphere, and your left hemisphere controls your right hand. So, if you have a severed corpus callosum and you hear a command to move your left hand, you will not be able to move your left hand, but you will be fine to move your right hand. So this situation is very specific to hearing a command, so a verbal command. If you just decide on your own, hey, I want to move my left hand, you do not have to get the information from the verbal command center. You can just use your motor cortex to be like, hey, I want to move my left hand. This area activates and tells the body to move the left hand. Okay, so person can still move the right hand even when you have a severed corpus callosum, and the person can still spontaneously move the left hand. The person can just not move the left hand when hearing a command or trying to follow a command. Okay, so this is the last slide for this video, and I will see you guys back soon for the next video.